This is the second set of slides from Lecture 8. This is the part about antibiotics. And in a lot of ways, we're going to think about antibiotics the same way we have to think about disinfectants. They have to be appropriate. We have to expose bacteria for the right amount of time. And we have to make them build up in the body to the appropriate concentration so they work. So all those concepts translate to antibiotics. And there are a few other concepts as well. And so this set of slides is not meant to teach you which antibiotics to use in which situation. That is a much bigger field, and there's a lot of really detailed knowledge you'd have to prescribe antibiotics appropriately. And that's not what an introductory microbiology class is for. Instead, this will give you practice thinking about antibiotics in the right way, and you'll start to be able to ask the right questions and understand why antibiotics were prescribed and what the differences can be between them. And this will be a good basis for if you want to go ahead and learn a lot more and get into the prescribing kind of mindset. So to start, again, we're going to look at some broader concepts, help you see what antibiotics are, and then we'll do some kind of detailed studies of a few antibiotics that will really show off the themes you need to know. So the first thing is, historically, what the heck are antibiotics and what did we do before them? This is a list that we can all chuckle about. We can all look at how clinicians used to treat bacterial infections back even before we had the germ theory of disease. And some of these make sense. One that makes sense to me is giving the patient a malaria infection. So they're infected by some unknown microorganism, infect them with Plasmodium and give them malaria. This causes severe fevers, and that is an innate immune response. And any pathogen trying to cause disease in your body while malaria is causing those fevers, any other pathogen has to deal with that powerful innate immune response. So this is better than nothing. Amputation makes sense if you have a gangrenous limb. We still do that today in some cases. Surgical debridement is still necessary in a lot of infection situations. But now we don't have to do therapeutic starvation or bloodletting. Those never made sense anyway. Now we can use antibiotics that directly attack the causative agents. So they directly attack the germs. Notice how often I use the word germs in this class. I don't. Germ is not a technical term. We just don't ever say it. I also never call bacteria or fungi bugs. You'll see people say, well, this bug is like this bug is like that, or I caught a bug and it made me sick. Call them what they are. Say microorganism, or say bacteria, or say fungi. If you know what they are, use the right term. That will elevate you in the eyes of other clinicians. Anyway, so back when we started really ramping up antibiotic production in the mid-20th century, so like in the 1940s and 50s and 60s, we were discovering new antibiotics, and a lot of infectious diseases became curable. And so that was really a golden age when people thought we were going to enter a post-infection era. But these days, 70 years later, we are looking dead in the eye of a post-antibiotic era where these drugs that have cured infectious diseases will disappear and stop working. So that's another thing I want you to understand through these slides is sort of what exactly does antibiotic resistance look like. So first we need to talk about some kind of essential terms that I'm going to use a lot, and you need to know what they mean. So we start with antibiotics because that's the big topic here, is antibiotics, and there are a lot of different definitions for them. I often say that they are any molecule we put inside us that damages microorganisms more than it damages us. And the idea there is that a lot of antibiotics are very toxic to us. So we can't say that they're toxic to bacteria and not toxic to us, because a lot of them are really toxic to us. So that's a thing to know. But for this class, a good definition is that antibiotics are they're chemicals that are produced by microorganisms that can harm other microorganisms. And what this does is distinguish them from antimicrobials as a whole. Antimicrobials would be agents that are capable of harming other microorganisms. So some antimicrobials are synthetically made. We found them in libraries of chemicals that chemists make and tested them. Antibiotics come from nature. Antibiotics were discovered in nature, and we may have learned how to modify them, or we may have found out how bacteria in nature make the antibiotics, and then we have copied that. We may have figured out how a fungus like penicillium makes penicillin. And in some cases, chemists can kind of use what we know to make the antibiotics faster, but in most cases, we still use those microorganisms to make the antibiotics for us because they do it the most efficient way and the fastest way. And so for a lot of times, making antibiotics requires growing a 3,000 liter culture of fungus or bacteria. Okay, so I will say these kind of interchangeably because I don't care that much about the distinction between these two. Other words that I'll use a lot that you definitely need to understand are resistant and sensitive. And this takes a little bit of practice, but I'm going to use them a lot. So it's important that you get this down now because there are a lot of hierarchical concepts in these slides. We build some big ideas and it's very easy to get lost along the way. And knowing resistant and sensitive will protect you from getting lost. So resistant is what we say when a microorganism can grow despite the presence of an antibiotic at its normal dosage or normal concentration. So we think about if you're going to use vancomycin, for example, to treat an infection in a patient, there is a typical dosage. There is a certain concentration that we can achieve in human tissues or in human organs of vancomycin before it becomes toxic. So if we use too much of it, it's going to damage the person. So there is kind of a maximum concentration we can achieve in human tissues before it becomes too dangerous. So if a bacterium can still grow despite that maximum concentration, we say it's resistant to vancomycin. So when we use a word like resistant, we are talking about a specific bacterium and a specific antibiotic, right? So we're saying this bacterium is resistant to this antibiotic. We're not saying this bacterium is resistant to all antibiotics. That isn't a real thing that ever happens. So the opposite of that is sensitive. We use antibiotics because we want them to stop bacteria from growing in our bodies. And so if it works, those bacteria were sensitive to that antibiotic. So, for example, if someone has a methicillin-resistant Staphylococcus aureus infection or MRSA, one thing we can use to treat that is vancomycin. And most MRSA strains are sensitive to vancomycin. That is, vancomycin can successfully treat infections caused by those MRSA strains. There are MRSA strains out there that have gained vancomycin resistance, and now they're called vancomycin-resistant Staphylococcus aureus because we have to know, don't try to use vancomycin to treat those. So what we want is the bacterial pathogens to be sensitive to our antibiotics. But what we face in reality is that many bacteria have become resistant to the antibiotics we treat them with. So why do microorganisms make antibiotics? This is a great question, and no one really knows the answer. I mean, the obvious first guess we would have is that the microbes are fighting against other microbes. But in nature, the concentrations of antibiotics are so low, they wouldn't work for this purpose. So even though we do have a few examples of these bacteria going to war against each other using antibiotics, in most cases we don't know why they make them. We're just glad they do. In Lecture 8, Part 1, we saw modes of action of disinfectants. And here we see modes of action of antibiotics. And if you take some time to look through this list, you can fit each of these into one of the modes of action that we saw in the other set of slides. And here's a diagram that can help you kind of remember the modes of action. And the ones we're going to look at in detail today are the cell wall synthesis inhibitors, which is the majority of antibiotics, and then the inhibitors of translation. So that's the mode of action. That spectrum of activity is another important concept. So think about these different groups of bacteria, for example, gram-negatives, gram-positives, chlamydias. So this is all the gram-negatives that aren't chlamydias or rickettsias. These are special because they grow inside our cells. They are obligate intracellular pathogens. They must grow inside our cells. And so any antibiotic we take to treat them has to get into our cells and achieve a high enough concentration to be toxic to these microorganisms. And as we will, well, I guess we won't get into it, but you can imagine differences between gram-positives and gram-negatives. To get into a gram-negative cell, a molecule has to get past the outer membrane in a thin layer of peptidoglycan just to get to the membrane. Gram-positives have a thick layer of peptidoglycan, and it's hard for a lot of molecules to get past that. They also have different surface carbohydrates and surface proteins. And so molecules are going to interact with gram-negatives and gram-positives and all these other ones differently. And that's why we can have a lot of different antibiotic molecules that have a different spectrum of activity. So, for example, penicillins. This is the oldest group, or the oldest major group of antibiotics, and they cover all the gram-positives. So something from the penicillin group would work against any gram-positive bacteria. That is before antibiotic resistance happened. But penicillins don't work against most gram-negatives, and they don't work against most chlamydias, and they don't work against any rickettsias. The tetracyclines are amazing because they can work against the chlamydias, the rickettsias, gram-positives, and most of the gram-negatives. The carbapenems are in some ways even more amazing. While they won't work against these two, they'll work against almost everything else that could bring a person to a hospital. So if you have, if you work in an ER and a patient comes in with a terrible infection and there isn't time to figure out what it is, one of these, there's a very good chance it would cover whatever is infecting your patient. So anyway, spectrum of activity is what we have to look at. And in each of these cases, these are large groups. Carbapenems is a group, the cephalosporins is a group, penicillins, tetracyclines, these are groups, not individual antibiotics. And so if you want to prescribe antibiotics, you have to know the details of every individual antibiotic drug you would be prescribing. And there are lots within these groups to know. So this table isn't what you would use in deciding what antibiotic to prescribe. This is just an illustration of the differences in spectrum of activity. So I think that's a good place to stop this video. I will see you at the next one.